Okay, hello everybody. Thank you for having me. I'm going to talk about what we do in the lab mostly. I'm a cell biologist. We don't do any math or modeling ourselves, but we collaborate with Bill Holmes here. But I have to confess right away that I understand very little in modeling. I can convey the conclusions that from the models, but if you have some detailed questions about how modeling is done and more detailed things to figure out, then Bill is here. So we study microtubules, and I study microtubules all my life and study it in different cellular environments, different scenarios. Today I want to touch on two stories in two different cell types where different microtubule network is necessary. So first off, just to, so do I have a pointer? I don't. Yeah, I'll do the mouse pointing. So we have, so I'll just have a few words about what microtubules are, in case some people don't know. Microtubules are protein polymers. They made of alpha-beta tubulin dimers as subunits, and these are self-assembling polymers which have two ends with different kinetics of assembly, plus and fast growing, minus and slow growing. And in cells, polymerization, depolymerization of microtubules is characterized by what is called dynamic instability, which results in fast switches between growth and shrinkage of those microtubules, which are stochastic. I'm not going to talk about these mechanisms. It is just a fundamental feature of microtubules, which is going on in all cell types. Importantly, the concentration of tubulin dimers in cells is so low that microtubule polymerization does not start spontaneously. So, because the minus end would depolymerize. So, microtubule polymerization in cells starts of templates, which is called gamma tubulin ring complexes. And this process is also called microtubule nucleation. That's where the original nucleus of the polymer is formed. And those minus ends are to be stable, and for microtubule to exist and not depolymerize, these minus ends have to be somehow stabilized, either by being kept by those gamma tubulin ring complexes or gamma Turks, or by some other stabilizing proteins. So gamma Turks are concentrated in specific locations in cells, which are called microtubule organizing centers, or MTOCs. And if it is a single microtubule organizing center in vertebrate cells, it is normally centrosome based. Then microtubules will radiate from that center with their plus ends toward the cell periphery. And the main function of microtubules to serve as tracks for intracellular trafficking. This trafficking is driven by proteins called molecular motors, and specific molecular motors can go either from minus end towards plus end, kinesins, or from plus end towards minus end, dynein. And this is a very schematic basis of microtubule-dependent transport. So for the transport in a cell to properly deliver cargoes from one location to another, two things are important: the geometry of the track and the activity of molecular motors. What we study in the lab is predominantly the geometry of microtubule tracks. So those microtubule networks in cells can be very different morphologies, and they're also dynamic. So they undergo dynamic rearrangements. The most dramatic one and the most famous one is rearrangement from interface cell, where most microtubules radiate toward the cell periphery, and bipolar mitotic spindle, which is illustrated in these two images. And I don't have my mouse anymore. However, this is not the only rearrangement. Even more spectacular rearrangement can occur during cell differentiation, because different cell types, especially specialized cell types, have also specialized microtubule networks. The best studied specialized cell type with, where microtubule network is very different is neuron. Down here, you can see that neuron has long processes, axons and dendrites, and microtubules are very different. These two types of processes. In axon, all microtubules are coaligned. In dendrites, they have mixed polarity, so they many microtubules face with their minus end toward the cell center, which is very unusual. Like another important cell type where microtubules have been studied significantly is epithelial cells. In columnar epithelia, in the upper corner over there, microtubules are vertical, which means that their minus ends are associated with the apical membrane, and their plus ends face towards the basal membrane. Doesn't you have a laser pointer by any chance? No? I will be waving my hands because I don't have a pointer, and this thing and the mouse has disappeared. Okay. Besides different geometries, it is important to keep in mind that microtubules are regulated by a variety of mechanisms. One is tubulin itself as a molecule can be modified through a variety of post-translational modifications, and also there are multiple proteins that bind to microtubules and modify their organization. Some of those proteins called their maps or structural maps. They can code microtubule along its whole length. Other proteins, plus tips, for example, are associated only with growing microtubule end. And there are also such factors which specifically destabilize microtubule by severing or by depolymerization. And these modifications can, all these modifications can change such properties of microtubules as lifetime, so the stability. They can also modify motor affinity. For example, if a microtubule is coated by certain maps, certain motors won't be able to walk along it. And post-translational modifications also influence motor affinity. And for sure, geometry. If a microtubule is curved, or if microtubules are bundled, or if microtubules are severed and relocated somewhere, all these things are regulated through different molecular mechanisms and define the architecture of microtubule network in the cell. But what is also important to understand is that the very first origin of a specific microtubule morphology could be actually nucleating microtubule at a different location. Because where microtubule is nucleated, the microtubule minus end is located, if nothing else happens. So the localization of nucleation position is important. And stop me if something doesn't make sense, please. So this has been one of our major interests in the cell, in the lab, historically, looking at where microtubules are nucleated. And this was one of the things that we found out when I just started the lab many years ago already, that microtubules can be nucleated at the Golgi complex, which is a big membranous organelle in the center of the cell. And we can follow microtubule plus end growth using this plastic marker, and we can track where the microtubules are coming from. And while tracking those growing microtubules, we can analyze where they come from. And they come from two locations here. One is this very radial array, which is centrosomal array, formed at the centrosome, this small microtubular organizing center that I was talking about. And another one is the Golgi, which is more scattered, but it is a distinct array. So in migrating cells, in motile mesenchymal cells, it is important that Golgi-derived microtubule array is asymmetric as compared to the centrosomal array, which is truly radial and extends equally to all sides. Golgi-derived microtubules extend predominantly toward the cell edge. And in a migrating cell, Golgi is located toward the cell front, which means that they are extending toward the protruding, leading edge of the cell. And this also means that majority of intracellular trafficking occurs toward the cell front. This is one of the basic mechanisms how cell can maintain directional migration by delivering all kind of material signaling factors, polarity factors to the cell front continuously through this extended or enhanced transport along microtubules. And over these years, quite a few molecular factors have been identified that are important for Golgi-derived microtubules specifically. And this is a very, very short and incomplete list of the factors that I have here. What is important for us to know is that gamma TORC, which is main template of microtubule nucleation, is necessary for Golgi-derived microtubule formation, and that also two other groups, Kia and Akmanova groups, have shown that gamma TORC activators are also important for Golgi-derived microtubule nucleation. And stabilization is also important. Anchoring of those factors to the Golgi membrane is also important. But right now, let's concentrate on just nucleation, activation of nucleation. In order to analyze the geometry of microtubule nucleation, we use the carousel washout assay, which is such an assay when we depolymerize all microtubules using a small molecule nacatazole. There is no microtubules in this cell, in nacatazole-treated cell, and the Golgi apparatus is fragmented because it depends on microtubule integrity. So these red dots in that cell that you can see are pieces of the Golgi. So when we wash the nacatazole off, microtubules grow back, and hopefully you can appreciate two big asters growing from the centrosome of the cell, and then a big number of smaller asters that grow from these small pieces of the Golgi, like right there. My mouse is back. See?
So that big aster, you're saying it's coming from the Golgi?
No, the big ones are from the centrosome. But yeah, but if you summarize all the microtubules coming from the small Golgi pieces here, it will be 50% or even over 50% of all microtubules in this cell type. So Annika Sanders, who is a postdoc in the lab, started looking more specifically at the geometry of formation of those microtubules in the carousel washout assay. What she has noticed is that even though the pieces of Golgi are pretty small, you can see that microtubules are formed in groups. We call it microtubule nucleation hotspots. You can see that it is not a single microtubule forming, but several of them. And in this piece of the Golgi, for example, there are two hotspots, one here, one here. So the whole Golgi membrane is not equal. It has some kind of locations where microtubule nucleation is favored. And this is non-random as compared to randomized simulation. So when we found it out, we needed to understand what are the rules of this nucleation. What could be the like laws that result in such non-random process? And this is why when we teamed up with Bill Holmes' lab, and he helped us to build a variety of models which could result in such a non-random nucleation. Three main models that were tested included cooperativity model, constraint cooperativity model, and heterogeneous Golgi models, and the two models that were found out to be consistent with the experimental distributions, experimental data, were two of them. Cooperativity model was the one which can be, like, again, in very lay words, as much as I understand the math, it's like the one microtubule is formed randomly, but second, third, and fourth are formed with higher probability at the same spot. But additional microtubules will be already added with lower probability, which means to me that the molecular mechanism of this hotspot formation is saturated at this point. And then there could be heterogeneous Golgi. So that means that a priori, before we even start nucleation, the surface of the Golgi was somehow heterogeneous and had the spots where microtubule nucleation can occur with higher probability. So from the grouping of microtubules, these two models were preferable, but when we analyzed the distribution of the length of the microtubules upon nucleation, the distribution of the length was more consistent with the cooperativity model. So, so far this our preferred idea is what's happening is that whenever a first microtubule is formed, it causes formation of additional microtubule through cooperativity mechanism. How could this happen? We have tested a variety of molecular mechanism, a variety of hypotheses, and I'm just, for the sake of time, I'm telling you about one that worked. And this is this activation of gamma Turk. So in order for gamma Turk to be active and nucleate a microtubule, it is thought that it has to undergo a conformational change, which is driven by so-called gamma tuna containing proteins, which again is a family of proteins. It is thought to be this kind of causing a closed conformation. That is, normally it is not completely a ring, but a little bit an open ring. But whenever it associates with this gamma tuna active domain, it closes up the ring, and then microtubule nucleation can occur. So if just this domain, gamma tuna, is overexpressed in cells, it results in formation of microtubules all over the cell, which indicates that gamma Turk itself is everywhere. It's basically dissolved in the cytosol and is only waiting to be activated. So any kind of specific formation of microtubules is rather regulated by activation of gamma Turk rather than just it being there. And then we also have an opportunity to use a mutant, which works as a dominant negative mutant of this domain, which significantly suppresses microtubule nucleation. And interestingly, it suppresses non-centrosomal cytosolic microtubule nucleation, but does not have such a strong effect on the centrosomal microtubules. So this works. So we have this quantification down here, which shows that the amount of Golgi-derived microtubule is significantly decreased when we express this dominant negative construct. But it is not completely lost. Actually, we don't have so far a way to completely eliminate Golgi-derived microtubules in cells. Whatever we tested, we don't have a tool that works all or nothing. But for this project, it was actually good because we still had some microtubules being formed, but their distribution only fit with the random model with the like no cooperativity model, as if microtubule nucleation occurs anywhere at the Golgi spontaneously, which means that when we block specific activation of gamma Turk, we block the cooperativity of microtubule formation. Microtubule still can form, so gamma Turk can be activated randomly or maybe by some other factors, but this is this gamma tuna containing proteins that have to get there in a cooperative manner. So this is the gamma Turk activation, which is the step of the process that works, that allows for cooperativity of hotspot formation. So how we think it might be working? So the gamma Turk and gamma tuna proteins, they exist separately in the cytosol. Whenever they get together spontaneously, one microtubule can be formed, and this allows for the feedback, which causes those proteins to get together, and this results in hotspot formation. We have several hypotheses how existing microtubule can cause activation of gamma TURC through gamma tuna, and we can discuss it if you want it during the questions.
So all of those proteins
Yeah.
are endogenously localized to Golgi, and see them there?
No, they are localized to the Golgi, but they're not enriched in the Golgi. That's the thing, that they are also in the cytosol. But in the cytosol, they don't, gamma TURC is there, it's not active. There's no nucleation there. So they have to get together to be active. And they work at low concentrations, clearly, because it is barely detectable by immunofluorescence. Now, getting back to organization of microtubules, again, remember I told you that Golgi-derived microtubules predominantly extend toward the cell front. So one of the reasons why this could occur is because the microtubules which are clustered, which are formed at the hotspots, they have have higher probability to be extended to the cell front rather than those which are random. So for whatever reason, which we don't know what it is, clustering of microtubules and formation of the hotspots results in a higher polarity of the Golgi-derived microtubule array. I have to say that it's been a few years since this polarity was discovered, but so far there is no good molecular explanation why Golgi-derived microtubules extend to the cell front and not random all directions. So it is possible that some kind of microenvironment in the Golgi periphery allows for higher concentration of gamma tuna containing proteins, for example. But this is this is total speculation at this point.
Do you have any thoughts as to whether that's a result of more nucleation on the side of the Golgi that's pointing towards the front, or whether it might be just more stabilization of microtubules?
So single microtubules are formed more randomly than clustered microtubules. So when they just simply nucleate randomly, it is not as polarized. So those which nucleate towards the front, they have higher probability to form a second microtubule. So yeah, that's all I know. So yeah, and the trafficking is again required for cell polarity and cell migration. So this is our little attempt to computationally analyze the directional arrays for delivery of factors to the cell front in migrating cells. But intracellular trafficking is important in many different systems, but there are some cells which do it for life. That's all they do. And one of those cell types is a pancreatic beta cell, the cell that secretes insulin upon glucose signaling into the bloodstream. And this is, as one can imagine, the extremely important cell type for human health and for the biology. And there is a lot known about those cells. However, the role of microtubules in insulin trafficking and secretion has not been explored very well so far. So we started looking at it a few years ago, and first we started off, and again, this is collaboration with the GU lab in our department, and again with Bill in terms of computation. So we started off just simply looking at the secretion process. How does the insulin secretion occur? It occurs in two waves. Once the cell or the pancreas receives the glucose signal, it secretes in two waves. Let me see if my mouse. No. So the first wave is our first phase of secretion. It's rapid, secretion of a high amount of insulin, and then there's this sustained or second phase, which is longer but less intensive. When people have been thinking about the role of microtubule in those two-phase secretion, they come up with their intuitive and very logical model in which the first phase does not depend on microtubules, because for the first phase, those insulin granules, insulin secretory vesicles, which are also called insulin granules, they're already located close to the cell membrane. So once they get the signal, they immediately secrete. So this does not require transport. So then there's the second phase, when more insulin has to be delivered from the cell center to the cell periphery for secretion, and this would require microtubule-dependent transport. So if this model is correct, then if we destroy microtubules, there will be no secretion in the second phase. First phase should still be there, second phase should be gone. So that was the understanding, and we started looking at it, and the first experiment that we did, we just simply destroyed microtubules in a cell, and as a control, measured the insulin secretion. Surprisingly, we got the opposite result than what we thought we will. So if you can see in that bar graph, the two blue columns is normal secretion, low glucose, high glucose, it goes up. But without microtubules, you can see that low glucose secretion is the same basal secretion, but the glucose-stimulated secretion goes up significantly, which is completely counterintuitive. And I always say that this was like one of those moments when I told Xiaodong, who did the work, it cannot be true, you probably swapped the tubes. But no, she didn't. So we had to retest this by a variety of means. One of fancy ways to look at it is to actually record single insulin secretion events. And this is a very nice assay, I like it, I'll show you the movie. This is a turf microscopy, which means that we are looking only at a very thin layer, basically at the cell membrane. And we use a fluorescein 3, which is a fluorescent dye, which only fluoresces when it is in complex with zinc ion. But this dye cannot penetrate in the cell. And insulin granules, insulin co-crystallizes with zinc inside the granule. So whenever it is secreted, local concentration of zinc goes up. So those flashes that you can see there, it is at the membrane. Whenever granule gets out, it gets fluorescent with fluorescein. So basically what this movie is supposed to show, that normally you have this much insulin secretion, but without microtubules you have this much insulin secretion. And the same thing can be tested by perfusion assay, which is more physiological assay looking in dynamics in whole islets. Whatever we tried, we found out that both first and second phases of insulin secretion are increased without microtubules. So this is not working. This model is not working. Right? So now, in attempt to understand how it works, I will very briefly go through, try to answer 3 questions. How microtubule network is organized, how insulin granules are moving inside those cells, and also introduce the model that Bill built for us, trying to understand how such microtubule network can result in this kind of insulin movement and localization. So microtubule network, how is it organized in beta cells? So this is microtubules in an intact pancreatic islet analyzed by super resolution microscopy, structural illumination. And you can see that the microtubules are shown here in green. You can see that this is a non-radial system. It is not this nice fibroblast organization. Everything goes with the plus and straight cell periphery. No, microtubules and beta cells are very complex, interlocked, dense meshwork. And insulin granules, which are shown here in red, are oftentimes constrained between these microtubules in all 3 dimensions. Now, we have analyzed this directionality. This was quite a bit of computational analysis, and like Dmitry has helped us to write this analysis algorithm, and Kangshiena and Kai have done the analysis. So this is how microtubules look in beta cell. This is a color-coded image. The green means microtubules which are parallel to the cell periphery, and the red is microtubules that are perpendicular to the cell periphery. The main conclusion out of it is that inside the cell, like right here, you can see that localization is random. All the angles are presented the same frequency. But at the cell periphery, there are many microtubules which are co-aligned with the cell membrane. So this is how this the cell looks. So this little thing in the corner there, it's like this peripheral bundle and crisscross pattern in the center. Just 2 words how the internal microtubules are formed, which has nothing to do with the modeling, but I just wanted to you to know that we did not disregard this interlocked pattern inside and analyzed it. These microtubules are actually Golgi-derived microtubules. They are nucleated at the Golgi in this cell type. This cell type, majority of microtubules are formed at the Golgi, which is pretty striking for such a specialized cell. So Catherine Trogdon in the lab has analyzed this. She has found that these microtubules are regulated by glucose. So whenever the cell receives high glucose signal, the amount of microtubule nucleation in the Golgi significantly increases. And she also has figured out the signaling pathway, which leads from glucose to microtubule nucleation and works through cyclic AMP and its effector EPAC2. So, yeah, this is a steady state movie which actually shows that this is an acatazole washout microtubules that are being formed and still image, and this is and the movie shows the similar thing in a steady state in the same cell before and after glucose stimulation. Importantly, what Catherine also found out is that these Golgi-derived microtubules in the cell interior are important for formation of insulin granules. Budding of membrane vesicles of the Golgi can be microtubule dependent in majority of cell types, or in some other studied cell types. It is not essential for formation of post-Golgi vesicles, but in pancreatic beta cells, it appears to be important, probably because of the sheer size of those secretory vesicles, which are much bigger than most. So basically, what she can see here, that without Golgi-derived microtubules, insulin is in the Golgi, but the surrounding cytoplasm is devoid of insulin granules because they cannot exit.
Is that just because they just can't move? They're just so huge that
we think that it is that they cannot. Basically, what is thought is that microtubules help to pull membrane tubes out of the Golgi, and then they are cut off by actin-dependent mechanism. That was shown in other cell types. There, it is a supporting mechanism. Actin can still do the job without microtubules. Here, they cannot do, clearly. We think it's because it has to pull a really big chunk of membrane with a lot of protein in it, which is probably very viscous. So that's why, but this is a speculation again.
I thought you said without microtubules there's more insulin secretion.
Exactly. Yes, I did. I did. So this, not in this case. So when we block Golgi-derived microtubule formation at the Golgi, we have less insulin secretion because, no, let me put it this way. The ability of cells to secrete insulin is not changed if we block just formation of microtubules at the Golgi. What is changed is the amount of insulin to be secreted in the cell. So the overall secretion goes down because there is no insulin to be secreted. But this is a long-term function. We don't have a tool that allows us to acutely block Golgi-derived microtubule formation. This type of dominant negative construct, it has to be expressed at least for 24 hours. And this is sufficient for the cells to be depleted of insulin significantly. So in long term, microtubules are required for insulin secretion because they are required for formation of vesicles. When I said that without microtubules, insulin secretion is increased, this is under the situation when insulin granules are present in the cytoplasm. And I'm just... It is a short term, yeah. I'll get back to it hopefully. Yeah. And I don't have I don't see the timer, so please let me know if I hope I'm not running too late. Yeah. But there is a second. So one thing that glucose does to microtubule system, it enhances this nucleation of Golgi-derived microtubules in order to make more granules. But another thing that we found is that the lifetime of pre-existing microtubules is decreased. So microtubules are depolymerized. So the whole microtubule network is remodeled. Old microtubules depolymerize, new microtubules polymerize. And we have detected this using this marker, glutubulin, which is basically a marker of microtubule lifetime. And we know that this destabilization is important. If we over-stabilize microtubules, secretion cannot occur. So Kang Xian is a postdoc in the lab who started looking at this microtubule destabilization process. And he used this photoconversion assay. This is a cell which expresses US tubulin, which normally fluoresces green. So he photoconverts a small area in the cell and then follows how fast those microtubules depolymerize. This is a pretty standard assay to follow microtubule depolymerization. Although with beta cells nothing is standard, everything is hard. So it was, and he did it in whole mount islets in tissue. So it was pretty hard, but. Nevertheless, what he found is that, yes, microtubules are destabilized in high glucose, but they only destabilized close to the cell periphery. Remember I showed you this distribution of directionalities? The microtubules which are close to cell periphery, they are parallel to cell periphery. And this is the area where high glucose causes microtubule destabilization. We have addressed how it can be, how it can be regulated. One of the potential ways to stabilize microtubules is to code them with those classic microtubule-associated proteins, structural maps. And we have looked at one of them. We have looked at several, but we ended up looking at one, which is protein tau, which many of you have probably heard of because it is one of the proteins associated with Alzheimer's, because it also stabilizes microtubules in neurons. But in these cells, it's highly expressed. And so to make the long story short, what we found, or Kang Xian found, is that this protein is sitting at peripheral microtubules. Hopefully you can appreciate that it is mostly the cell periphery, and then there is a quantification for that. The gray line shows localization of tau to microtubules in low glucose. So you see it's higher in the beginning. But in high glucose, it's gone. So he also found that glucose causes phosphorylation of this protein. Then it falls off microtubules, and this is a standard process which has been described in neurons previously that allows for microtubule destabilization. So what happens that in low glucose, tau is sitting at peripheral microtubule, stabilizes them, but in high glucose, it is falling off, and those microtubules now can exchange fast. So this is what we know about microtubule network. more or less, in pancreatic beta cells. So what do we know about insulin granule transport? As I mentioned already, there are plenty of plenty of insulin granules in a cell. So you can see that they are filling the cytoplasm. Basically, this electromicrograph. You can see many granules in the cell. And from the statistics of the insulin granule numbers and the amount of insulin secretion each stimulus in organisms, it has been calculated that only about 4% of granules have to be secreted at each stimulation, which is less than the amount of the granules even sitting beneath the membrane in those cells. Which means that if we secrete all the granules which are beneath the membrane, it will be too much. And oversecretion of insulin is very bad because it causes hyperglycemia and can result in coma and death. So this is one important thing. Second thing that we found, actually consistently with other data in the field, that insulin granules do not move direction of those cells, which is not surprising if you think how convoluted the microtubule network inside those cells is. So the movement of those granules follow the subdiffusion law. Whether microtubules are present, the blue lines in low glucose or high glucose, or if microtubules are not present even, they all move in a subdiffusion mode, only microtubules just make this movement faster. And another important observation that we made is that when we destroy microtubules, we can see this results in the slow down of insulin granule movement, but it also results in accumulation of an excessive amount of insulin granules on the cell periphery. So if you look at those numbers, no mouse. If you look at those numbers, you hopefully can see that normally in control in the MSR, the amount of granules which arrive and which are withdrawn from the cell surface is the same, or approximately the same. But the amount of granules which arrive in the catazole, which means no microtubules, is lower than normally because they move slower. But then they cannot be taken away efficiently, and will result in accumulation of a high amount of granules at the cell border. Interestingly, this non-significant difference between arrival and withdrawal that we see in control cells is the same as the number of secretion events per cell that we see. It is a not significant difference, but it correlates very well with how many granules have to be actually there to secrete. But if we look at what happens in a catazole, way fewer granules secrete than how much is accumulated. So they accumulate, and there's still an additional mechanism that regulates how much we'll secrete. Still, we secrete too much. These numbers show that we secrete 2 times too much, which is on the physiological scale, it is a lot. So this is this movie again, which shows how we quantify the amount of secretion. So there are 2 levels of regulation of secretion. One is this balance between withdrawal and delivery by microtubules. And another one is an additional regulation, which actually regulates the exocytosis of available granules, which probably involves those mechanisms of exocytic machinery, including snare proteins and calcium-dependent activation of exocytosis. What we observe in the catazole allows us to hypothesize that without microtubules, there are too many granules allocated at the cell periphery, and this allows for extra anchoring to the membrane, and the results to extra secretion. Right. So this was our hypothesis, and if in this scenario a microtubule is destabilized, then under normal microtubule dynamics conditions, for example, high glucose causes microtubule destabilization. In this particular spot, microtubule is destabilized, so a granule has a chance to attach to membrane and be secreted. Okay, but how can we coordinate this unusual microtubule network with this peripheral bundle and this non-directional transport, which results into actual withdrawing granules from the cell periphery? And this is where Bill's model comes in, and he built a model in order to answer two questions. One is how this accumulation of granules in the cell periphery regulated by microtubule transport under these conditions, and also what's going on with this membrane binding, membrane docking for exocytosis, how this is regulated by microtubule network transport. So the model involved generation of this in silico cell, which has microtubules being generated inside of the cell, and those which reach the membrane, they can cover, they turn and curve around it, forming this bundle. And the insulin granules are distributed randomly throughout the cytoplasm, which is a starting point in a steady-state beta cell. The model accounts for the granules can move along microtubules, they can diffuse in a microtubule-independent manner, they can also bind and unbind to microtubules, and they also can bind and unbind to the cell membrane, which is normally stimulated by high glucose. And for sure, all of these conditions were modulated this or that way by the model. So the main conclusions are, which is that in general, the presence of microtubules by itself leads to enrichment of the granule cell periphery, which is not surprising because originally the granules generated in the cell center, they have to get to cell periphery somehow. But this microtubule network, which is aligned at the cell periphery, this peripheral bundle that we see, it results in accumulating peripheral granules and taking them away from the cell periphery. Another important conclusion out of the model was that the result that we observe with granule accumulation in the periphery in the absence of microtubules, this can only occur under the conditions when the docking of the granules at the periphery is activated. Without this, without the activation of this mechanism, there will be no accumulation at the end. And only those granules which are not bound to microtubules at the moment should be able to dock and eventually secrete in order to be consistent with our experimental data. So whenever granule is bound to microtubule, it is taken away. So if the model allows all granules to anchor and secrete, then the data don't correlate with the experimental data anymore. Which means that loss of microtubules would promote insulin granule docking, because these granules will become unbound to microtubule. And when I say loss of microtubules, in our experiment, it is loss of all microtubules, but in physiological conditions, it could be loss of microtubule in just this, in a particular site close to the membrane in the process of normal microtubule destabilization, high glucose. This is already like my interpretation. So if you want to know more about the models, there's more. Because there's we have the paper has been recently accepted in by a physical journal, so there's more details to that model. So if you have any questions, ask Bill. So these are the main conclusions from the model, though. One is that microtubule alignment is the crucial mechanism needed to withdraw granules from the periphery, and that microtubule transport not only positions granules within the cell, but also controls whether they dock to the membrane or not. And making a step back into the molecular mechanisms that I described a few slides ago, this tau-dependent microtubule stabilization should be regulating not all these things, right? Because this what regulates microtubule stability and local microtubule depolymerization. So tau would promote microtubule stability and alignment close to the cell membrane, so it will promote withdrawal of granules from the cell periphery. And when tau is removed from microtubules depolymerize, this is what would allow for granule binding to the membrane and secretion afterwards. And this highlights the importance of the tau-dependent regulation of insulin secretion. And that's all I have. These are my guys. By the way, Bill and I are looking for a joint postdoc. If you hear of someone who is looking for a position which involves both modeling and wet lab, let us know, and I can take any questions.
I have an initial question. So I guess your model is very interesting because it's sort of the opposite way of how I think of vesicles docking into the membrane. I usually think of them being delivered on microtubules, and then they find a docking site, and then they can dock and release. And instead, you're saying they're sort of finding docking sites, and they're being picked up and pulled away.
Even if they're delivered by microtubules, which they are, only in a non-directional manner in this system, but in order to efficiently be docked, they have to be first detached from that microtubule, which we think occurs when microtubule depolymerizes. That they're delivered, that microtubule depolymerizes, and then it can dock.
One kind of going back and forth, back and forth, back and forth, and boom.
Yeah, because I think that even if, from the mechanical point of view, if a granule is being pulled by a motor on this parallel bundle, like moving fast, even if the docking molecules attempt to bind it there, this binding might not be enough to overcome the force that the motor exerts on the granule. And it is very low probability that this connection will be very stable. That's how I see it. Yeah?
Maybe this is obvious, maybe you addressed it and I missed it, but earlier on you had this picture of like the cell and the microtubules were aligned by the edge and not so much in the center.
Yeah.
What is the reason for that again? Is it like a structural thing, like they can't point this way because there's a dimension?
Yeah, so the best answer to this question is we don't really know how it happens, but we think that there's how it can happen is that first off, microtubules are nucleated in the cell interior on this fairly big organelle, which is Golgi. So they already don't come from the cell one point. They come from a variety of nucleation sites inside. And then as long as they grow toward the membrane, as long as they hit the membrane, they are so stable that they don't stop and they don't depolymerize back. Rather, they turn and start, making kind of circles around it. This is one possibility, which was also incorporated in the model. and this is something that has been described in other cell types, this kind of turning behavior and growing along the cell membrane. But an additional mechanism could be that already polymerized microtubule is being transported toward the cell periphery, and instead of growing and turning, being transported and turned and sit there as a shorter microtubule, which is probably a part of the process. We have an ongoing project where Kai is a graduate student who looks at this transport of microtubules that detach and move. But there might be other additional mechanisms that remodel the microtubule network so that it starts looking like that.
Is it possible to, if you think the transport dynamics are partially responsible, to like stop, if you think it's being transported,
Yeah.
is it possible to stop the transporter from working to see how the alignment changes?
Yeah, yes, we're going to look at that. We're going to look at that. So Kai has already determined at least one molecular motor, which is kinesin 1, conventional kinesin, which was implicated in microtubule sliding in other cell types. It definitely contributes to microtubule sliding in beta cells, and we can deplete it and look at microtubule network. It's going to happen soon, hopefully.
So I have a question about plus versus minus end. So was the direction of how the cargo was transported regulated? Also, if you
Yes.
mentioned like the insulin granules were somehow blocked by microtubule blocking, so
Yeah.
presumably they were transported back or something. But as you mentioned, if the microtubule were coiled in a way, then the plus minus end were all
Yeah.
So was there maybe some other mechanism that pulling or guiding those granules back to the center?
Yeah. So in the model, we assume that granules can be moved by both minus and plus end direction motors. And it has been shown that both kinesin and dynein can move a granule. In the old models where people thought microtubules are radial, it was always assumed that it is kinesin plus end direction motor that delivers granule to cell periphery. But we know that both kinesin and dynein can move it. So which motor is predominant in this situation? It's an open question. Actually, I can tell you that we actually have some results by kinesin depletion, and I don't know what it means, but it results in enrichment of insulin granule cell periphery. Kinesin is plus end directed motor, supposed to deliver to plus ends. When you get rid of it, something is broken in the balance of this non-directional movement, and we get enrichment at the cell periphery. No idea what it means yet. We need more modeling to understand it. But yeah, it's complicated. And there are other kinesins which in beta cells, which people have not looked at all. So normally people just consider this kinesin 1, conventional kinesin, to move insulin granules. There are other kinesins expressed. We don't know their roles. So, no answer.
How available are the docking sites? I know that was in the model as well, but is the number of docking sites also regulated by glucose, or are they there all the time and it's just regulated by depolymerization of the microtubules?
In the model, we don't have predominant docking sites, right?
They're absent when glucose is.
Yeah, but otherwise they're random, right? Anywhere they are or not.
The docking propensity does have to be very weak, though, because if docking is a strong propensity thing, it overwhelms everything in the system.
Yeah.
So I mean, if you have a high affinity docking, then nothing else you do matters in the system.
Yeah.
So it has to be a low affinity.
Yeah. Yeah. But there are those high probability docking sites. in beta cells are known, these calcium channel dependent hotspots of secretion. And we actually have some preliminary data in another project which indicates that without microtubules, there's still those preferred docking sites. But in some cells secrete, some cells don't secrete normally, because these beta cells are heterogeneous in the islet. Some cells are active, some are not active. When we destroy microtubules, it seems that each cell becomes active, but it still has a designated docking site. But that's, yeah, we still need to get good statistics on that, and yeah. No more question? Thank you.